let's go over chapter 10. Let's go here. Here we go. All right, so the big thing here, looking at the respiratory system, is really just to get the basics on how this whole thing works. All right, so when we're talking about respiration, we have four things to think about, and the first one is just breathing. And that is breathing or ventilation is what we call that. That's just moving air in and out of the lungs. External respiration is what we think about when we have the exchange of gases between the lungs and the blood. Whereas internal respiration, that is when we have exchange between the tissue and the blood. All right. And of course, cellular respiration, right? This is the whole reason we're taking in or we're ventilating, and why these two things happen, the external and internal respiration, is so that we can have cellular respiration, which we covered way back, I think, in chapter 2, right? So this is where we use oxygen to make ATP, and that takes place in the mitochondria using the Krebs cycle and the electron transport chain. And of course, we started with glycolysis, but we don't need oxygen to do that, but we need to have glycolysis to feed into the Krebs cycle and the electron transport chain. So I put the equation up on the board just to remind you about the fact that this is involving the two main gases that we're considering here, right? So that's, and you can see on page 1 of my notes, so just ADP plus phosphate plus oxygen and sugar, right? Oxygen on the substrate side, and on the other side, products, we're giving off waste, right? CO2, which we then breathe out, as you know, which is why we're talking about this. We make a lot of water, which we then pee out, which we'll talk about when we do kidney. And then, of course, we get ATP, right? Now, the respiratory tract is divided into regions, and I was saying in class, depending on what book you're using, they divide it in different ways, but the book that we're using is divided into upper and lower, all right? So the upper respiratory tract, there's a lot of stuff going on there, right? We have our sense of smell. We have the fact that we're warming and moistening the air that comes into our bodies, because as you know, our body is mostly water and some carbon. That's mostly what we are. And because of that, we can't have this dry air coming in because we dehydrate ourselves. So we need to warm it and moisten it and warm it because we have a warm core temperature of 37 degrees centigrade. So we have to warm up that air, especially in the winter here, right? We need to filter out all the particles that we inhale. When we inhale a lot. When I was living in Manhattan, I lived right across from a coal-powered electricity generating plant, and there was so much soot on everything. So thank goodness for the hairs in my nose when I went for my run in the morning. But I digress. Anyways, the other thing is our voice, right? And the transport of gases, because clearly we have to get those gases deep into the lung, where we're going to have the exchange, which is happening in the lower respiratory tract, where we still have transport, and finally the exchange of gases, because that only happens deep in the lungs of the alveoli. Now, I went over the parts of the system here, the anatomy, right? Just basic anatomy. If you move on to AMP, you're going to learn a lot more details here, but the nose and the mouth are ways that we get air in. Of course, the mouth is also for food and liquid, but and they join here at the back at the pharynx. And there's 3 parts to the pharynx, but here we just talk about the pharynx. There's different names for the part of the pharynx that's behind the nose, the part that's behind the mouth, and the part that's going into the larynx here. But that's the pharynx. It's a double passageway for everything that we take in this way. So air, food, and liquid. And I also was mentioning that we have in the nasal cavity, we have these conchae, which increase the surface area, allowing for the warming and moistening of the air and filtering, because it's all covered with hairs. All right. Then the larynx, this is the top of the voice box, and I'm going to go over that, but the epiglottis is part of that. It's one of the 9 cartilages that are there, covers up the larynx and the entrance into the trachea when you're swallowing. So this epiglottis, it folds down when we swallow, and then the food goes down the back here, and the liquid down the esophagus. Then, of course, we have the trachea, which is otherwise known as the windpipe, but please call it the trachea. We're in physio here, human bio, that's the proper term. And then we have two bronchi, one for each lung. And then they further divide into bronchioles, one for each lobe. And I'll get back into that, but we have 3 lobes on the right lung and 2 on the left. And we'll talk about that. And then we have other things that are important for breathing. So our diaphragm, the muscle is the main muscle of breathing. And then we have other muscles, internal and external intercostals that help us to take in more air, but I'll go over that in a sec, all right? And then deep in, we have the alveoli. We're actually having the exchange of gases, and then we have many, many, many iterations of these branches, which are called bronchi. All right, so let's keep going here. Now, I wanted to remind you that each lung is in a body cavity, which we discussed that way back, I think chapter 1 or chapter 2, I can't remember, but we have body cavities. And remember, we have the pericardial cavity around the heart. This is the mediastinum here, which is sort of a space in the cavity. But we also have the 2 pleural cavities, and as such, they're surrounded by the double serous membrane, the internal of which is called the visceral pleura and the outside, the parietal pleura, all right? And in between these layers, we have the fluid that keeps because it is a serous membrane, so it's making that watery fluid, that keeps this all supple, and it reduces friction as the lungs expand and contract. Then I went on to the larynx, and there we've got 9 cartilages. You don't need to learn all 9 of them through this course, but that is how we get this larynx, which is also called the voice box. And why is it called that? Well, because this is where we have our vocal cords, or our vocal folds, and they are in the center of the larynx, and this is where we get our voice. And you can watch videos of how the vibration of those control our voice pitch and how that works. And you'll see in the videos that we have muscle contractions here that change the shape of the resonating chamber, the larynx, for the voice. Right? And then part of this is the epiglottis. So epiglottis is one of the 9 cartilages, and it covers the opening, which is called the glottis. All right, so if someone asks you to point out the glottis, you can say it's just an opening, so there's nothing there, but there is a name for that opening, and it's called the glottis. And you can see some of the other names of different cartilages here, but each of the 9 cartilages has a name. You can see here too the thyroid cartilage and the cricoid cartilage. And the reason I brought that up is because if one needs to insert a trach or a breathing tube, which is called a cricothyrotomy, and the reason it's called that is because the tube is inserted between these 2, the cricoid and the thyroid cartilages, into that space there, into that connective tissue. And this is just the thyroid. That's what this is here. And we'll talk about that when we do hormones. Okay, so that's pretty much all I want you to know about the larynx. All right, okay. Again, we have different numbers of lobes in the 2 lungs. So the right lung, there's 3 lobes. In the left lung, there are only 2. You don't need to learn the names of the lobes for this course. But they, of course, have names. Each and everything in anatomy has a name. So, you can see that we've got different lobes. Not that they're hard to learn: superior, middle, and inferior, and just superior and inferior. That's, you don't need to be a rocket scientist to figure that out. But you can see that. And the other thing here is that the left lung has slightly less capacity, and that is because the heart, as you know, is slightly left of center, and the apex of the heart sort of fits right here into this cardiac notch. Even though the heart is in its own chamber, it does affect the shape of the chamber of the left lung. All right. Okay, next thing, the trachea. So the trachea, you can feel those cartilages. They're C-shaped, or I think it looks more like a horseshoe, but horseshoe-shaped cartilages. Your book calls them C-shaped cartilages. I know that so many of you are in the equine program, you can appreciate that being a horseshoe. I don't know. Anyhow, that's not why I use it. It just looks like a horseshoe to me. But the point is, it's not a circular cartilage, and that's really what one needs to keep in mind here. It doesn't surround the entire thing. On the side of the esophagus is when you swallow, it will push in on the trachea, which is one of the reasons why it's very difficult to breathe. If you get something stuck in your esophagus or something is having a hard time to get down there, it will block the breathing tube, right? So here I am using that term, but block the trachea because it is soft enough that it will come in here and block this. Now here it's a very muscular membrane here, right? And we can control the diameter of the trachea somewhat using smooth muscle. But one of the reasons we have these cartilages is to keep all of these airways open so that we can breathe, right? But there is, of course, remember way back I was talking about the smooth muscle, circular and longitudinal smooth muscle around any and every, lumen or passageway that we have in our body, and it does include the trachea, all right? So all of the respiratory passageways are surrounded by smooth muscle, which will also affect the size of those passageways. Okay. Now, the other thing I want to note is that the trachea is lined by a ciliated mucus-secreting epithelium. It's a pseudo-stratified ciliated one, and I'm going to show you a picture of that and explain again why that is so important. And I also use this slide to show you the location of the cough reflex, which is right at the end of the trachea where it branches into two bronchi. And this is called the carina, all right? So we have sensitive cells here that will detect any type of particulate matter and will initiate the coughing reflex. Now, this epithelium, the reason it produces mucus is because in between the epithelial cells, we have those secretory goblet cells. Remember, goblet cells are the ones that secrete mucus. So they're here, and here you can see the cilia. So it looks kind of hairy, right? So anytime you see this, you know that you're looking at airway, all right? You can see it's pseudostratified. The nuclei are all over, even though these are columnar cells. This is not transitional. It is pseudostratified columnar. Now, if you look at it here, these little hairs, they beat. And I was saying, so beating meaning that they're moving, like they're waving, they have a purpose. And what they do is that the lower respiratory tract, so down in the trachea, these cilia are going to be beating upward. So any particulates that get stuck in the mucus that is sitting on them, okay, will get pushed up towards the larynx, because we're eventually going to swallow those, right? So once it gets out past the larynx and into the pharynx, we will be able to swallow it. And the same thing for any ciliated epithelium above that point, they will also beat, but they will be beating towards that, so beating downwards. So again, so that we can swallow any sort of particulates that get stuck in the back of our throat and in our nose and all of that. So it won't go down into the trachea, it will be swallowed. So remember that the cilia beat in different directions depending on whether you're north or south of that dividing line, so that you can swallow those particles. Now in class, if you weren't there, what I had drawn was the epithelium, and I drew the cilia, and I drew the mucus. And I wanted to highlight the fact that the mucus is actually floating on a layer of watery fluid, okay? So I want you to remember that. We're going to talk about the importance of that shortly, okay? Now, looking at the cough reflex. So when the carina initiates the cough reflex, two things are going to happen, all right? So first of all, the smooth muscle is going to tighten the passageway, so it's going to get narrower. And the other thing that's going to happen is that air is going to be forced out from deep in the lungs, out through the contraction of the abdominal muscles, and it's going to force that air out at an increased rate. And with the smaller diameter, because of the contraction of that smooth muscle, we have a lot of force to push any particles out with the cough reflex. Now, in a healthy airway, The cilia, like I said, are going to be beating to move things that, so that we can get rid of them. But what happens with a smoker, there's a lot of toxins and particles from the burning of the cigarette that enters into the lungs and into the airway passages. And what that ends up doing is causes a lot of irritation. And that irritation causes an increased buildup of mucus to trap all of that extra sludge that's coming in there. I'll be honest, it's basically sludge. And with time, the cilia get destroyed with all of that extra mucus and the damage from the toxins. So it really destroys that buildup not the buildup, it destroys that part of the cellular makeup of this epithelium, because it just can't be sustained, those cilia, under these conditions. So then in order to clear the airway, smokers need to have the coughing, right? So that is that violent coughing that particularly heavy smokers will encounter, because they have basically destroyed the mechanism that the body uses to expel all of those toxins. And then you can imagine a lot of these toxins get into the lungs, and they also cause irritation in the lungs and mutation, and because of the increased cell division. We'll talk about that shortly, but that leads to lung cancer. Okay, now the other thing I wanted to highlight here is in a situation where we have this increased mucus buildup. The other one is for cystic fibrosis. Now, the reason this happens for someone with CF is because they have difficulty producing that aqueous layer between the cilia and the mucus. And the reason they do, and if you remember my saying, and I keep harping on that in this course, water follows salt, right? Or water follows solute, but it's usually salt when we talk about the body, because of osmosis. So what the body has to make that sort of aqueous layer is a series of pumps or channels that pump salt out into the lumen, and that draws with it water. And what happens with someone with CF, they have a reduced number, or they do not have one of these transporters, which is called CFTR. And it's not that they don't have, it's not functional, okay? There's a mutation that destroys the function. So they don't get that nice liquidy layer between the cells in the cilia and the mucus. So the mucus sits, and it's very difficult to clear this mucus without that aqueous layer, similar to a smoker, right? But someone with CF does not have all of those toxins, and it's not the same mechanism that leads to the buildup of the mucus, clearly. But that mucus just ends up sitting there, and they have a regimen of clearing that mucus daily, which is quite exhaustive. There's videos on YouTube. You can see what someone with CF has to go through to help clear this, and that's just so that they can breathe. And the other problem is the buildup of the mucus. There's going to be increased rates of respiratory infections, because, again, they can't clear out the bacteria that starts settling in that mucus as well, which is also the reason that smokers have an increased rate of respiratory infections as well. Okay, hopefully that makes some sense to you. Now, with the buildup here of the branching of what goes on after one gets through the trachea and the bronchi and into the little bronchioles eventually, as these passageways branch, they get smaller and smaller and smaller. And this up here just sort of shows you that after you have the two bronchi, then there's going to be one branch per lobe, and then it's going to continue. And the reason that this is happening is so that we can create a very vast surface area for the exchange of gases, because this is just happening by diffusion. So the more surface area we have, the more efficient the exchange will be. So by the time we get to the actual organ of exchange, which is the alveoli, which I'll talk about in a sec, we have a huge surface area. We have more than 1 million square centimeters, all right? And I said in class that I read somewhere that that's bigger than the area of a football field. I really don't know, because I don't know how big a football field is. I'm sorry, I'm a total science geek, football and me, we don't get along too well, but anyhow, so maybe one of you athletes can check that for me. I don't know the size of a football field, but this to me, but the point is it's an important visual. If you can imagine that surface area is inside of you to allow for the exchange of gases. And that's why I like to use it, because I think we could all, even though I don't play football and I don't know much about it, I do have a strong visual as to the size of a football field. So I think it's something we can all relate to. Okay. Okay, and I'm assuming that that football field is not referring to soccer, which is also big. And I know that a lot of you in my class think that real football is what we call soccer here, and I'm not going to debate you on that. But the point is you get the visual, all right? Okay, this is what happens when I do a video on a snow day. I digress. All right, let's move on. Okay, now the alveoli. Now this is where we have the exchange. Now, not only do we need a really large surface area, but we also need to have very few barriers to that exchange. So it has to be very efficient. And as such, we have a very, very thin barrier, cellular barrier, okay? Because we have a surface, so we have an epithelium. We have an epithelium on both sides. Okay, so of course, on the alveolar side, we have a simple squamous epithelium. So that respiratory epithelium has changed from that from that, oh my gosh, I'm playing with my earring while I'm talking. It changes from that ciliated columnar, pseudostratified appearance, right, with the cilia all the way down now to a simple squamous flat epithelium, all right? So there is this transition that happens between the trachea and the alveoli to that epithelium to change the morphology, all right? So don't think that we have that pseudostratified columnar ciliated situation in the alveolus. We do not. All right. And that, of course, since it's an epithelium, is sitting on a little basement membrane. It's very thin. And then we have the blood vessels, which, of course, as you know, have also that simple squamous characteristic, right? And we call that an endothelium. It's very special. Remember where that simple squamous epithelium is called an endothelium when we're talking about blood vessels or lymphatics. All right, so that's all we have for the gases to get through, right, in a normal situation, which I will expand upon shortly. Now, so that's a very thin layer for exchange. So we have this huge surface area and a very thin membrane. Now, those cells that make up that simple squamous epithelium, they're called type 1 cells, and they're devoted, of course, to gas exchange. But we also have two other types of cells in the alveoli. The other type is called the type 2 cells, and they produce something called surfactant. And surfactant is like a soap. It's a sort of a very sort of a slippery liquid, okay? And why do we have that? And in class, I tried to use the analogy that if you have a balloon, or if kids, you know, the kids, when they always, when they get their hands on a balloon, they like to blow it up and, you know, let it shrink again and blow it and shrink it. And, you know, eventually they're chewing on it and it gets to be a sticky mess, right? And then it gets harder and harder to blow up. And the reason that is because there's so much spit in that balloon that the surface tension, remember, water, which is in your spit, has a very high surface tension. It likes to hold on to itself. That's why, if you remember that image that I had of the water moving up the tree, right? It pulls the water up the tree from the roots to the leaves. It's holding itself together, right? Now, remember, one of the jobs of the nose is to moisten the air that we take in. So that air that we're breathing is moist. So it gets into the alveoli. There's a lot of water in there. So the same thing would be happening to our alveoli if we didn't have the surfactant. It would start to stick together and they would be difficult to inflate. So that surfactant there makes it sort of that slippery substance so that the alveoli don't stick together every time we exhale and they contract, right? Or they shrink so that they can open up again when we inhale, all right? Now, hopefully that makes some sense to you as to why we have them, because if we don't, we get sick, and I'll talk about that shortly. Dust cells, those are macrophage. We have macrophage that live in our alveoli, and they're called dust cells, and their job is to clean up stuff that we inhale, right? And bacteria. And remember, the macrophage used to be those blood monocytes. Remember, I had said that all of the leukocytes or the white blood cells can leave the bloodstream and return, except for the monocytes. When they leave, they become the macrophage of that tissue. And those macrophage for the lungs are called dust cells. Okay? If you recall, the other macrophage that we've encountered so far are the ones in the skin, which are called Langerhans cells. And we'll encounter more as we go through the course. Okay, so then once we get into deep into the lungs, we have exchange, right? So we have the big surface area, we have the close contact, and this allows for the efficient exchange of gases, carbon dioxide and oxygen, right? The only other time we worry about another gas, which is the nitrogen, which actually is most of the air we breathe, is if you dive, because that's all of a sudden if you put the body under pressure, nitrogen will become soluble in blood. But at normal pressures, nitrogen is not a factor. Okay, so if you're a diver, that's why you have to be careful, all right? Because nitrogen is bad for you, okay? Now, how does this all work? Well, there's two things that are at play here, okay? So gases are going to flow down a pressure gradient, okay? And that pressure gradient can take two forms. One of them is literally pressure, right, that you think of, but the other one sort of is the concentration of those gases, which we refer as the pressure of each of those. So it's a little confusing. But first of all, let's consider the overall what we would normally think of as pressures. And the way this works is that the muscle of breathing, the diaphragm, okay, in a relaxed state, that muscle rises into the chest cavity, and you can see that the space is smaller. But when we inspire, the muscle contracts and it goes down, and that increases the space of the thoracic cavity. When you increase the space, you decrease the pressure, so that draws the air in. Okay, so the thoracic cavity is a vacuum, right? Which is why if it gets punctured, the lungs collapse. Okay, and the hole has to be sealed up pretty quick. Now, then when we exhale, the muscle relaxes. So expiration is actually a passive process. We're just, relaxing the muscle. And the space gets smaller, the air is pushed out. Okay, so I think this makes sense, right? The size of the cavity, the bigger the cavity, the lower the pressure, the air gets pulled in. Okay, now, and the diaphragm is really the muscle of breathing. But of course, we have other muscles that we can expand the thoracic cavity even bigger than when we're relaxed, or squeeze it in even tighter. And that's when we recruit other muscles to do this, right? So other muscles for inspiration, of course, diaphragm. But the external intercostals, you don't need to memorize. Hopefully you'll just remember this. But the external intercostals, and then for expiration, the internal intercostals and the abdominal muscles. All right, so these are the ones that will be involved. And if you're really going to get further into it, we have the sternocleidomastoids and the scalenes, which will also help pull this rib cage upwards. Okay, but it's usually these around the rib cage that we think about here, but they can be recruited. Now, when we're talking then about the capacity of the lungs, right? This can be recorded using a spirometer. Now, what we're looking at here is the regular breathing. This is your diaphragm breathing, is the tidal volume. And you can see it here on the graph. It's your regular inhalation and expiration. And it's just sort of, the diaphragm expanding and relaxing. Contracting and relaxing, contracting and relaxing. And this is sort of your resting breathing rate. But then if you recruit all those other muscles, that's your reserve volume. So you have an inspiratory reserve, okay, and an expiratory reserve, right? So if you recruit all of those and you really get that chest cavity, I find myself doing it while I'm describing it, but you get the chest cavity even larger and then shrink it down even tighter. Then you bring in your reserve, and that is when we have the vital capacity. So the tidal volume is the regular inhalation and exhalation in a single breath, but when we have the vital capacity, we've recruited in the inspiratory and expiratory reserves, and that is now giving you even more air to exchange. And when we're looking at these, we're really thinking about where we're actually having gas exchange down in the alveoli, right? Now, you're never going to squeeze those alveoli all completely together. And that last little bit of air left in the lungs, even after you've used your expiratory reserve, is called the residual volume, all right? So there's still going to be a little bit out in there. Then finally, the dead space volume. So this is the volume of air in the airways that isn't involved in exchange, right? You have all of that cartilage in your trachea, keeping those airways open. And I didn't mention this now, but I'll mention it now. But as we go through from the trachea to the alveoli, you get progressively less cartilage, but everywhere where you have cartilage, those passageways will remain open, right? Because the cartilage, even though the smooth muscle will move in and out, it will not completely close down. So that's your dead space volume. All of that space where you're not actually having air exchange, it's always going to be filled with air. So that's the dead space. Okay, and it's not shown here on this graph. Okay, I'm not even looking at my notes. Hopefully I'm not missing anything. Let me just flip through. Okay, so now we're going to talk about the actual diffusion gradients, all right? So we've talked about, high to low pressure in terms of the space, right? But we also high to low pressure in terms of the concentrations, right? So this is slightly different, but it's a pressure gradient nonetheless, all right? So what we're talking about here is the actual concentration of the gases, which is expressed as a pressure because it is a gas. So if you look at dry air, right, the total pressure of all the gases in the air, including oxygen and CO2, is about 760 millimeters of mercury. This is at sea level, okay? Where we're at here at the school is less because we're above sea level. But the point is some of that is oxygen and some is carbon dioxide, and about 160 is oxygen and 0.25 is CO2. Now, think about this, right? So let's go down to the bottom where we have the tissues. Now here, the pressure of oxygen is less than 40 millimeters of mercury, and the pressure of CO2 is greater than 46. So look at the difference, right? So we have 160 to 40. Now it's diffusion. It's going from high to low concentration or high to low pressure. This is going to draw air that we take into the alveoli, into the blood. Let's put it on this side because we're talking about oxygen, into the blood, and then out to the tissues, and then into the tissues, right? That concentration gradient is going to keep pulling the oxygen inward. Conversely, look at the CO2. We're starting here at 46 millimeters of mercury, and here we have less than 1, like a quarter of a millimeter. So now this is going to be the opposite. That difference is going to draw, that concentration difference is going to draw that CO2 out of the body, okay? So hopefully that's making some sense to you. And you can see how these pressures change at the different places, so internal and external respiration here, okay? All right, so we have those two aspects, right? The concentration or the partial pressures of each of these gases and the space of the chest cavity. That's all working here for you to help us in the breathing process, okay? All right, okay. All right, so now how are we transporting those gases? So oxygen. So oxygen is transported in two ways, right? So of course you know at this point that oxygen is bound to hemoglobin. We get oxyhemoglobin. But you have to think about this. We have the alveolus, and we have the blood, and then we have the red blood cell. So that oxygen has to go into the blood and then into the red blood cell, okay? Now remember, the plasma is mostly water, right? I think we have like 92% water. Now, the thing is about oxygen is that it doesn't dissolve well in water. So only less than 2% of our oxygen is actually transported, dissolved in the plasma. The rest of it is bound to hemoglobin, okay? So that would be your oxyhemoglobin. Now, remember here at the lung, I was talking about the shape of that hemoglobin protein. Now, at the lung, we have a neutral pH. Why do we care about pH? Because it affects the shapes of our proteins, right? So we have a neutral pH, we have low temperatures, and we have a high pressure or high concentration of oxygen. And that is all conducive to allowing oxygen to bind to the hemoglobin. Now, conversely, at the tissue, right, it is more acidic, and I'm going to get into that in a sec. It's more acidic. That's going to change the shape of our protein. It's going to be less, our hemoglobin protein, and it's going to be less inclined to bind to oxygen. Its affinity for oxygen will decrease, which will help unload it, which is what we want, right? We want to unload that oxygen on our tissues. So that acidic pH is going to help change the shape of the protein. We also have warm temperatures, so that's going to speed up that exchange. And then again, of course, we have that low oxygen concentration, so that's also going to passively pull that oxygen off the hemoglobin into the plasma and then into the tissue, right? So remember, the plasma is the barrier here. It has to go through that to the tissues, okay? All right, now, hopefully that makes sense to you, all right? So we can see what's happening. Now, CO2, right, our carbon dioxide is a little bit different, okay? First of all, CO2 is quite soluble in plasma. It's more soluble in water than is oxygen. So we have three ways that we carry CO2. Now let me talk about the first two first, because I think they make the most sense at this point in the course, right? So the first one is just dissolved in the plasma. So 10% of our CO2 can dissolve in the plasma, and it's just carried as CO2 in plasma. Now, I had mentioned to you before, remember that we've changed the shape of our hemoglobin at the tissues, right? And we have a lower pH. And remember I said that when we were talking about blood and hemoglobin and red blood cells, that CO2 can also bind to hemoglobin. And at this point, it can be bound to the hemoglobin, and we have carbaminohemoglobin at this point, okay? Don't get confused with carboxyhemoglobin. That's carbon monoxide bound to hemoglobin, which I told you is a toxin, right? It's poison. But here we have carbaminohemoglobin, and 20% of our CO2 is transported back to the lungs in this form. Now the other 70%, now this is a little tricky, but so important to the body, all right? Our CO2 is converted into our biological buffer, right? All right, so bicarbonate, all right, HCO3 minus, okay? This is our biological buffer, and I mentioned this way back in chapter 2. So important to our body because it buffers out all the acid, all right? We need to keep our body pH at a certain level, and that is done in the plasma at least using, and you'll see other places, using bicarbonate. So how does this happen? How does that happen? It's really strange, right? How do we convert that CO2 to bicarbonate, which then is going to be put into the plasma? Now, it's done using an enzyme, right? Remember, enzymes are so important to us. It's done using this enzyme called carbonic anhydrase. Now, where is that enzyme? Well, it's actually in the red blood cell, all right? So that's what the point is over here. So CO2 is going to come into the plasma. Some is going to stay like that, right? Some of it is then going to diffuse into the red blood cell. And as I mentioned, 20% of that is going to stay bound to hemoglobin. But the other 70% is going to go through this reaction that's catalyzed by carbonic anhydrase. It's going to combine with water, okay, which we know we've got lots of, right, in the blood. And it's going to make bicarbonate. This bicarbonate is going to be transported out of the red blood cell, okay? Now, notice we are making some hydrogen ions here. Okay, so this is why, this is why our body pH is more acidic in the tissues, because we're making this conversion for the CO2, and part of it is making this hydrogen. that can be neutralized somewhat using chloride, okay? So some of it is neutralized using chloride. It's kind of a weird sort of situation to think about, all right? But we neutralize a little bit by making the salt, okay? Of course, it can dissociate, all right? That does happen. But I want you to keep this in mind, all right? So if we build up CO2, we're also going to be a buildup of this acid, all right? And this is why at our tissues, the pH is a little bit acidic compared to elsewhere, okay? So that's really important to keep in mind here, okay? Now, when we get back to the lungs, right? Now, this is when that concentration gradient takes hold. It's going to start pulling the CO2 that's dissolved in the plasma out of the plasma and into the alveolus for expiration. But in so doing, it's also going to be pulling the CO2 out of the red blood cell, right? It's going to be pulling that off of the hemoglobin because now we've changed our pH and everything is changing. And it's also going to allow for the reverse reaction. So carbonic anhydrase is going to catalyze the reverse reaction as well. So it's going to take that hydrogen, okay, and it's going to convert it now into water and CO2. I'm going to get rid of that extra chloride, all right? So in the long run, it all buffers itself out, but this is where we're making our, our conversion here, and that happens at the lungs, all right? So I know it's a little bit of a complicated process, but it works, and it's so critical to our body, and the enzyme here is carbonic anhydrase. Where is it found? It's found in the red blood cells, okay? Okay, let's see here. Make sure I'm not missing anything. Do a quick look at my notes. Dissolved in plasma, carbaminohemoglobin, acidic pH. Okay, there we go. And when you take AMP, if you're one of those students, you will learn a little bit more about this chloride thing here. Okay? All right, cool. Now, how do we control breathing? There's a few ways that the breathing is controlled, right? So first of all, we have stretch receptors in the alveoli that sort of make sure we don't overexpand our lungs. So we have that, okay? So these stretch receptors respond to the amount of stretch in the muscle and in the alveoli, and they will stop the expansion that signals the brain. But let's go through this here. We have a respiratory control center. Where that's found is in the brain. The location is in the medulla oblongata, which we'll talk about when we do brain. We're going to do that shortly. And it responds to several things, all right? So it controls several things. So first of all, we have receptors near that location that respond to changes in hydrogen ion concentrations, right? Now remember the equation, right? CO2 on one side, as we build up CO2, we're going to get hydrogen, all right? So in that way, these receptors are actually responding to increases in CO2 because of the byproduct that's made via carbonic anhydrase, that hydrogen. And that hydrogen ion concentration diffuses from the blood into the cerebral spinal fluid, which is detected by those receptor cells in the medulla oblongata, okay? So that is one way, so we respond to the concentration of CO2 or increasing concentrations of CO2 by corollary by increases in hydrogen ion concentration, which diffuses into the cerebral spinal fluid. Now, the other thing that we have, of course, are receptors, and we have them again in the carotid artery and in the aorta. So carotid going to the brain and aorta going to the body, respond as well to decreases in oxygen concentration in the blood, okay, which of course would make sense, right? So not only do we have pressure receptors here for blood pressure, but we also have receptors that sense decreases in the concentration of oxygen. So if we have decreased concentration of oxygen and increased concentration of acid, that's going to signal our respiratory control center to increase our rate and depth of breathing. So increased CO2, increased hydrogen, and decreased O2 will all converge upon the respiratory control center and say we need to increase our depth and rate of breathing, okay? And that is going to be a parasympathetic response. We will talk about that shortly. Then, of course, we have conscious control over respiration too. I can tell myself right now, take in a deep breath, or the doctor will say, breathe deeply. You have conscious control of that as well, and that happens in the cerebrum, okay? All right. And as you know, the diaphragm isn't smooth muscle, it's skeletal, so you also have control over it, okay? Okay, so hopefully that's making sense. Now let's make sure that we've covered everything. Yes. Okay. Now, normal lung. Okay, let's talk about things that go wrong, right? So normal lung, the distance between of that membrane in the alveoli is about 0.1 to 1.5 microns, all right? It's really thin, right? Remember, micron is to the -6th of a meter, right? So if you remember in lab, we used the meter stick, and those little teeny tiny lines on the meter stick, the tiniest ones were the millimeters. So a micrometer is 1,000 times smaller than that, right? So it's really, really small. And we're talking about, you know, a 0.1 of 1,000 times, so it's like, you know, 10,000 times smaller than that, those lines, okay? So normally we have a very close contact between the alveoli and the blood, okay? So we have really efficient exchange. And was there anything else I wanted to mention? Oh yes, and don't forget the surfactant is going to allow for those alveoli to expand easily, no matter how much surface tension from the liquid that builds up in there, it's still going to allow for that exchange. Okay, so now let's see what can go wrong. Emphysema. Okay, so this is a characteristic disease of the smoker, all right, and of people who have suffered damage due to toxins in the lungs. So what happens here is that the alveoli are destroyed. Okay, so we actually damage the alveoli, and when that happens, that builds up scar tissue. And importantly, it decreases the surface area that we have for exchange, okay? And this is that, and if you've seen that, there's a terrifying commercial of someone dying of emphysema on the television, and basically they feel like they're suffocating to death. And this is why, because you've lost, or that patient has lost the surface area needed for exchange. And I don't know how much of that it is versus that football field analogy, but enough that the person feels like they're suffocating. Okay, and this mostly results from smoking, and they have a shortness of breath. So this is what's happening with emphysema. So the destruction of the alveoli means less surface area for gas exchange. Now, pulmonary edema. Remember, edema is buildup of fluid around spaces, okay? So pulmonary edema is a buildup of fluid around the lungs. And remember, that was one of the things that happens with congestive heart failure. The heart is pumping less efficiently, fluids are starting to build up, okay, because things are just not working right, okay? And what ends up happening is two things. First of all, we have an increased distance between the blood and the alveoli because we've got this barrier of fluid. And remember what I said, oxygen is poorly soluble in water. So this makes it even harder for that oxygen to traverse this distance. We can get the CO2 out because CO2 can dissolve better in water, okay? But you have a decreased oxygen carrying capacity in the lungs, in the blood, because you can't get as much oxygen into the blood because of, first of all, the liquid and the increased distance build up there. So this is what's happening with pulmonary edema and what's happening to someone with congestive heart failure. They have trouble breathing. Okay, asthma and bronchitis, all right? So what's happening here is a couple of things. So first of all, with asthma, there's contraction of the smooth muscle, and that's going to constrict the airway. So it's going to be harder to get more and more air into the alveoli. Even though the alveoli are fine, you're going to have the constriction of the airway, all right? So the smooth muscle in asthma is constricted, and it also happens with those allergies as well, right? So this is controlled by bronchodilators. Now bronchitis, right? If you have bronchitis, which I feel like I sound like I have bronchitis right now because I've had this chest thing going on. What's happening there is not the smooth muscle, but we have inflammation, and that's going to restrict the bronchi. So it's going to have a decreased oxygen carrying capacity. We'll see how my run goes tomorrow with my restricted airways here. But that's what's happening there, okay? All right, so hopefully that makes sense. Now, the next one, respiratory distress syndrome. Now, this brings me back to our type 2 cells and the surfactant production. Now, what happens to someone with respiratory distress syndrome is that they need to be on a ventilator. Why? Because those type 2 cells are not producing surfactant, or we don't have enough type 2 cells, and the lungs get progressively difficult to inflate. So the ventilator is going to force the lungs to expand. And this is very, very common in preemies because they just haven't developed yet to the point where their type 2 cells are active and producing surfactant, which is why preemies very often are on the ventilator. And some of you know, but my daughter was a preemie. She was born 12 weeks early. And I digress again, but I was so worried that she wouldn't be able to breathe when they delivered her. So I think the happiest sound that I ever heard was her scream, because that meant that she could expand in her lungs. And so that was a good thing, because ventilators are so dangerous. I mean, she was on the vent because she was so early. But the danger here for preemies is that it causes blindness. So I was total tiger mom and trying to constantly turn the vent down. I know, but it sounds terrible. But I didn't want her to be blind. So everything turned out fine. You might, I don't know, I'm doing this on Wednesday. You might see her on Friday because she's Friday off, but she's fine. But she didn't have those type 2 cells to produce the surfactant. And as such, she was put on the vent after a couple of days because she was just laboring too much for her breathing. She was one tough cookie though. Anyway, so that's what's going on there. Now, respiratory infections. We've got a whole slew of these, right? So tuberculosis. You just have to learn this stuff. This is a bacterial infection, and please do learn it because it's on the exam, all right? Make sure you know these. This is like four or five questions on these, identifying these different situations. So tuberculosis, bacterial infection, and why it's so damaging to someone's respiratory pathways is because the infection actually scars the lungs. And it can be diagnosed with the skin tests and chest x-ray. Treatment is antibiotics, all right? Very contagious though. Botulism, this is a poisoning by a bacterial toxin, and it causes paralysis of skeletal muscles. Why is that important for respiratory pathways? Well, that diaphragm, remember? Skeletal muscle, as well as your intercostals. So you lose the ability to breathe because this toxin basically paralyzes your muscles. Colds and flu caused by viruses, so therefore antibiotics are not effective. Okay, so please don't go to your doctor asking for antibiotics for your cold and flu. It's just causing a whole antibiotic resistance thing that's going on. So it won't work. Pneumonia. That's the infection of the lungs. This can be caused by bacteria or viruses, okay? And it is one of the top 10 causes of death in the United States. And the treatments can depend on the cause, right? So viral, you're not going to be able to take antibiotics on that. Bacterial, yes. Now, the reason this is so dangerous is because it's usually happening to patients who are already compromised and they just can't breathe. It just gets terrible. But anyway, so pneumonia. And this is an infection deep in the lungs, as if you've ever had it, you know, all right? Now, the last thing is lung cancer. Many types of causes, smoking is a big one, All right. So 90% of the cases are associated with smoking. Other things, such as radon gas, up here in upstate New York or central New York, whichever you call it. I was living in Manhattan for so long that up here is just like the boondocks compared to someone living. It sounds weird. I know I'm from Toronto, but anyhow, radon gas is a big problem here. And in my home, I have a radon gas system that sucks it out of my basement and pumps it out to reduce the possibility of getting cancer. But basically, when you buy a home in this area, they do a radon gas test, and they look to make sure that the levels are lower than allowed. Then workplace chemicals such as asbestos, so emphysema. Not emphysema, mesothelioma. That's what I was thinking of. Mesothelioma. Emphysema is from smoking, but mesothelioma is the asbestos-related cancer. And this is, again, due to the irritation of those chemicals in the lungs, that causes mutations because the lungs are constantly trying to repair things. Anyway, so what happens? Uncontrolled growth of abnormal cells. It impairs air flow, gas exchange, blood flow. Hopefully by this point you understand why that's all important. And it accounts for 1/3 of all US cancer deaths, right? So how do they treat it? They can try to get rid of parts of it surgically. And I just put this in here so you can imagine. So they can do like a little localized part or a whole segment, an entire lobe, or a full lung, which is a pneumonectomy, okay? All right, and that is chapter 10. Chapter 11 is coming, all right? So hopefully that will clarify everything. The snow day this week has completely messed things up, and I know a lot of you, we're not back on Monday, but hopefully this will help you. And Friday, I will go over things quickly so we can catch up because we do have that midterm next week, all right? All righty.